Thank you very much. Very happy to be here. Oops. No. Okay. So my name is Ben. I'm from the Weizmann Institute. I'm a PhD student in Uri's lab. And I'll be talking about our recent work on reassessing the heritability of human lifespan. So when you look at when we look at the human traits from twin studies, human traits tend to be around 50% heritable. This is from a meta-analysis done on thousands of studies. So across different functional domains, cardiovascular, endocrine, immunological, so on, we tend to see around 50% heritability. This is relatively well known. And what's interesting is that in the 90s, studies began coming out about the heritability of human lifespan based on twin studies, Swedish and Danish twins. And they found around 20%, so half that. And even more recently, large-scale family tree data, Calico study and another study published in Science, found even lower estimates for heritability, putting it at around 10%. And the plot thickens when you consider similar estimates in mice. So in the year 2000, on four-way cross mice, genetically heterogeneous mice, they found heritability that's roughly 50%. So when you look at just this forest plot, I think it's clear that something is up. And it's kind of interesting to see what's going on here. So when we look at the most cited, the most well-known study on the heritability of lifespan in humans, it's on the Danish twins. They were born in the end of the 19th century. And we have here the monozygotic and dizygotic identical, non-identical twins and their correlations. And combining them together using the well-known Falconer's formula, you have the heritability estimate. But what's interesting and what I want you to point out is obviously these guys over here. What is this? We have twins here where one sibling lives to be 80, 90, 100 years old, and the other dies at the age of 20, 30. This creates a very strong off-diagonal effect that seeks to lower the correlations, and it's something that nowadays I think is quite strange to us. So to understand where this is coming from, it's useful when we look at mortality curves in human data. The well-known Gompertz exponential phase is usually what's called intrinsic mortality. And usually from the age of 20 to about 40, we see a plateau in human mortality that can be attributed to extrinsic mortality. This is work done by Olshansky and Carnes. And if you look at, you can plot this extrinsic mortality over time. This is in Denmark cohorts. And you can see that the value of the extrinsic mortality from the 19th century to today has dropped pretty much tenfold. So in the 19th century, it was about 0.4% to die each year. That may sound small, but if you do the calculation, it means that your chance of dying from age 20 to 40 is about 20. So 20%, 1 in 5 people died from 20 to 40 by that 0.4%. And nowadays, obviously, it's much, much lower. So now let's try to conceptualize this. So how do we put this together? So we can think about let's say people with a particular genetic basis as belonging to a genetic group. So just for demonstration, let's take 3 genetic groups here. And if we plot the correlations, there's a symmetry between 2 twins. So we have this kind of symmetric cloud distribution. And one can show mathematically is that for an arbitrary amount of genetic groups, the correlation would be the variance of their means divided by the variance of their means and their spread. And one can show mathematically, and indeed we do, that what happens is with extrinsic mortality seeks, by including early deaths, it compresses these distributions together, and it also widens them. So it does both things. It both messes up, it lowers the variance of mu, and it increases the sigma. So both effects actually serve to lower the correlations. So this is just putting some math on the, I think, very intuitive understanding of what extrinsic mortality does. This is more like just a simulation where we take, let's say, 3 different Gompertz, let's say 3 different Gompertz groups, on the left without extrinsic mortality, on the right with extrinsic mortality, and you can see that the extrinsic mortality compresses them together and also adds a short tail there. So hopefully I think I've convinced you that what's the interesting question is obviously the heritability of lifespan due to intrinsic mortality. That is the biological data, that is the signal. The extrinsic mortality is the noise, and that's what we are after here. So in order to do that, what we do is we introduce heterogeneity into mortality models. So we take two different mortality models. We take the SR model, which Uri and Ifan talked about, and we take another model, which is just a parametric fit, the Makam-Gamma-Gompertz model. And the idea is that now each person will have a particular parameter based on a distribution. And we take a Gaussian distribution to reflect polygenic additive models. So just additive genetic traits. That's, by the way, 70% we think of human physiological parameters are additive. And using that, we can then simulate twins and identical twins and non-identical twins. So identical twins, we would say they have identical parameters. This is the symmetry between them. They belong to the same distribution. And the non-identical twins would have 50% correlated parameters reflecting the 50% genes that they share. And using this, we now have a tool where we can now simulate these genetic groups, which is what we do. So here, for example, we did a simulation that captures the heritability seen in the Danish cohorts, and we see also these lots of individuals on the, points on the off diagonal. But the power of the simulation is that now we can easily distinguish which are extrinsic and which are intrinsic. So the red dots are the deaths which are extrinsic, and we can see that they're very strong on the off diagonal, which again screws up the correlation. And now because we have this simulation, we can also remove them and be left with just the signal. And the signal to us is we find roughly twice the amount of heritability that's previously known. Also, I want you to notice, this is very important, that a lot of the heritability is because of these dots down here, that there are twins who are dying at age 40 and 20 because they have really poor genes. But in the historical cohorts, you can't tell if this is intrinsic or extrinsic. We don't have, the data doesn't include the cause of death. So our simulations allow us to kind of really probe this, I think, much better. So we can also do a plot where we can do the heritability versus the level of extrinsic mortality. And obviously we see that at high extrinsic mortality, it's very low, and it pretty much plateaus at a level even higher than 50%. I'll also mention that we tried, like I said, two different models, but the results are relatively the same. We just wanted to see that our results are model independent. And now we can do something interesting. We can plot the same curve, but we can do it for different cutoff ages. What do I mean by that? So now I can do the same thing, but where I only include twins where both live to be a certain age. So age 20, 30, 40, and so on. And then we get this interesting effect where when extrinsic mortality is very high, obviously increasing the cutoff age increases the heritability, because what I'm doing is I'm throwing away people who died due to extrinsic reasons. I'm throwing away noise. But when extrinsic mortality is low, the opposite happens. When extrinsic mortality is low, actually a high cutoff age reduces heritability, because now the noise has become signal. Now people who are dying early is because of their bad genes. And now when I'm throwing them away, I'm messing up my estimates. And we also compare to 3 different studies. We have the Danish twins, the Swedish twins, and SATSA. This is new data from our collaborators in Stockholm, Sarah Haig's lab. And we can see that our models pretty much capture quite well, I would say, the different correlations. So let's zoom out a minute and be kind of maybe a bit more philosophical about this. So I think it is important to realize heritability by definition depends on the environment in which it is measured. So by definition, it is the amount of genetic variance divided by the total variance. And therefore, if we were to measure heritability of mice in a trap-infested house, the heritability would be low, but mice in the lab would be high. And I think that exactly is the same for humans, although we can't grow them in a lab yet. So what we're interested in is, we would want to control the same environment and to reduce extrinsic mortality, and then the heritability that we would measure would ideally be just down to genes and this inherent stochasticity. And in that sense, I think the recent studies, it's not moving, can you help me? Yes, one more? So the recent large family tree studies that found 10% heritability They weren't wrong, of course. I think it's just the question is what was the environment that they were measuring? And they were measuring an environment across different centuries with a very low cutoff age across different countries. So sure, I mean, their estimates were correct for their data, but I don't think the question they were answering is the right question. I think the question that actually is interesting for us when we're talking about the heritability of lifespan, can you help me one more, is the intrinsic stochasticity. And that's what our simulations and our kind of modeling approach allows us to do, to disentangle the intrinsic aging and mortality from the extrinsic. One more. Yep, and that's it. I just want to thank our collaborators from Stockholm, Thais and Sarah, and of course from the lab, Ifan, Glenn, Nave, Avi, and Uri, of course, and we'll have a preprint out soon, hopefully in the coming weeks. Thank you.
Have you done any investigation into the shape of the curves you get under large-scale intervention trials, such as caloric restriction for mice? Does it change the shape of the curve for heritability?
You're talking about heritability in mice?
Well, I'm just thinking in terms of the if you're removing extrinsic mortality. So now you've got, say, a mouse study, like an ITP mouse study, and you're doing some intervention like caloric restriction. And you're removing all those extrinsic factors. Do you maintain that same expected shape as you would for just wild-type mice living out their life and, without interventions?
So regarding to my knowledge, there's only one study that I found on the heritability of lifespan in mice, and they regressed parent to offspring regressions. I didn't see anything done with caloric restriction or with interventions. Maybe you're asking, like, if I understand you correctly, how would interventions impact these correlations in general?
Yeah, basically.
Yeah.
And there have been some fairly large intervention trials around things like caloric restriction where you're removing all the other factors, right? So I'm just wondering if you think of it, is the survivability or is the lifespan just moving up the y equals x-axis at that point with the intervention?
Yeah. So I think it's an interesting question. In general, I would say with this approach, one could say that if I were to know if both twins were exercising or not exercising or eating well and so on, so we would get different. Right, I'm actually kind of assuming no priors. When I'm assuming the symmetry between twins, it's kind of saying, right, I just know that they're twins and all the rest I don't know. And when I if I add priors, I think I could, it would change the picture. So I think this is maybe like the kind of zero order kind of estimate for this kind of thing. But it's interesting to think about things.
Thanks.
Okay. So I have a question about the longevity, whether they are heritable or not. So I'm not sure whether it would be exactly matched with the centenarian studies. So I'm not sure whether this kind of stuff only can be found in centenarian groups or not.
Yeah, so that's interesting question. Thank you. We have another work that I thought I was going to talk about, but in the end I decided to talk about this. And we actually we exactly answer that question to try to see what are the relative longevity advantages for different ages. So let's say if I have a sibling who's 100 years old and so on, and we see that indeed we can reproduce many of the centenarian longevity advantages. We also see that the relative survival probability, for instance, if your sibling or your twin lived to be a certain age, what are your chances of living to that age? We see that the excess survival probability actually rises exponentially. So that's something that has been seen empirically, but I think we can show that using our simulations that theoretically we expect it to rise exponentially as well.
Okay, thank you.
So you made me think about the heritability of longevity differently than I have, so thank you for that. That was great talk. So one of the things I'm wondering is, the environment has changed a lot, as you alluded to, in the last 100 years, 50 years. So extrinsic mortality has come down, but we're also seeing an obesity epidemic and a healthcare system that's keeping sick people alive longer. And so I'm wondering a couple of things. One is, could you look at the heritability of health span, to whatever extent we can measure health span. I think that would be interesting. And then maybe speculate a little bit on the impact of obesity and you know, the environment that we live in today and how that might play into this.
Yeah, those are very interesting questions. I think this work was primarily focused on, you know, taking the well-known kind of, let's say, textbook data and cohorts on this type of work and trying to disentangle signal from noise. And that's the simulations, that's what we were trying to do. We were trying to recreate this kind of Danish cohorts, both correlations and mortality statistics, but to be able to differentiate between which deaths are extrinsic and which are intrinsic. So you're suggesting something very interesting. I think that would be taking it a step further for, let's say, modern day cohorts, for example. And I'd be happy to talk about that.
Cool, thanks.
Let's thank all the speakers for this session again, for the wonderful talk.